Good day, everyone. Today, I, Gabriela Nicole Cipre, together with my partner, Eliza B. Gonzalez, will be discussing about designing primers for restriction-based cloning into a target plasmid. This presentation will walk you through the purpose, the principles, and the full workflow for designing cloning primers that are accurate, functional, and compatible with restriction, digestion, and ligation. So what is cloning? Molecular cloning is a technique used to insert a DNA fragment, often called an insert, into a plasmid vector. This allows us to express genes, mutate them, purify proteins, or study gene function. Restriction-based cloning specifically uses restriction enzymes to cut the vector at defined sites and insert our gene of interest. To make this work smoothly, we designed primers that not only amplify our gene but also add the restriction sites we want to use for cloning. So for the workflow overview, here's the general process when designing cloning primers. First is you need to identify the gene sequence that you will amplify. Second is to identify the multiple cloning sites or the MCS of your target plasmid. Third is choose two restriction enzymes that are present in the vector but absent in your gene. Fourth, design forward and reverse primers with clamp, restriction site, and gene-specific sequence. Lastly is to check the temperature, the GC content, and the dimer formation using tools like primer tree or benchling. So for designing forward and reverse primers, during the lecture series of Dr. Angelo Banyares, our DOST Balik scientist, one of the key topics he discussed was the proper design of primers for molecular cloning. When designing primers for restriction-based cloning, the forward primer is built with a simple structure: a clamp for efficient cutting, followed by a restriction site, and then the gene-specific sequence that usually begins with ATG to ensure proper expression. The reverse primer follows the same pattern but instead contains the reverse complement of the gene after its own clamp and restriction site, commonly using a different enzyme site. Using two different restriction sites ensures directional cloning, making sure the insert goes into the plasmid in the correct orientation. Now for the clamp region, most restriction enzymes do not cut efficiently at the very end of a DNA fragment. To solve this, we add a 5 to 6 nucleotide clump upstream of the site. These can be random bases, typically GC-rich for stability. Without the clamp, the enzyme may fail to digest your PCR product and ligation will not work. Now let's have the tools for primer design. First, we have benchling. It is typically used for plasmid maps, restriction sites, and auto-generated primers. Then we have APE or a plasmid editor that is lightweight, offline, and great for MCS checking. And then NEB cutter that identifies restriction enzyme sites in DNA. And then primer tree that calculates primer stability, temperature, and secondary structures. And lastly, we have SnapGene Viewer that is for free and is used for excellent visualization of plasmids. Now let's go through the key guidelines for designing primers, especially for cloning. First, primers generally need to be 18 to 24 base pairs long. This length is ideal because it gives good specifics without being too long or too hard to bind. And then second, aim for a 40 to 60% GC content. This helps maintain stable binding between the primer and the template. Next, it is recommended to start or end with 1 to 2 GC bases, also called as GC clamp. GC bases bind more tightly, so this helps the primer attach more firmly at the ends. And then the melting temperature or TM should be between 50 and 60 degrees Celsius. Also, your forward and reverse primers should have temperature values within 5 degrees of each other so they bind at the same time during PCR. And Another important rule is that your primer should not have complementary regions. If they do, they might bind to each other and form primary dimers instead of binding to your DNA. And for cloning applications, you normally add 3 to 4 extra nucleotides at the 5' end before the restriction enzyme site. These extra bases improve enzyme cutting efficiency. And then also, try to avoid secondary structures like hairpins or strong internal loops which can interfere with proper annealing. And then, avoid long repeats such as 4 or more of the same base in a row or repetitive pattern like 80 80 80 because this can cause slippage during PCR. And finally, avoid homology within the primer or between primers, meaning don't let more than 3 bases complement each other inside the same primer or between forward and reverse primers. Too much complementarity leads to self-dimers or cross-dimers, which reduces PCR efficiency. And those are the essential guidelines to follow when designing primers for cloning. And now let's proceed with manual designing primers for restriction-based cloning. So for the first step, you would like to get first your insert, and in this case, we will be using PEGFPN1. So on your desktop, open NCBI GenBank and find PEGFPN1. Then click FASTA and copy the sequence. Now pasting it into a different sheet, let's now design the primers. So first, note that we will be getting the first 20 nucleotides of our insert and we will label it as our forward primer. Next, the 20 last nucleotides will be our reverse primers. In order to design it, you still need to get the reverse complement. You can do it manually or you can open the site for automatic reverse complement as I'll show you now. Now that we have our forward primer and our reverse complement, let's now proceed with designing them. Don't forget to insert the GC clamp and the restriction site. In this case, we will be adding ACAR-I as our restriction site for the forward primer. So it should look like this. Next, we will be adding the GC clamp and the HIND3 on our reverse complement. So it should look like this. And that is how you design your primers for restriction cloning. Once done, we have to download our target plasmid now. You can also find it in the same site if you'd like. Copy the sequence. Next, there's this site called NE Builder. Just pick the orange button that says Add Fragment. Once done, you need to process text. Then check vector if it's a vector, and plasmid if it's a plasmid. And then click or type the name of vector. Then click Restriction Digest. And then now you can insert the 5 and 3' flank of your insert site. Once done, click Add. Now insert your new fragment and introduce the new sequence. Once done, you need to click PCR and copy both your forward and reverse primers. Now, as you can see here on the right side, this is now our final results in which we were able to insert our restriction-based clone on our target plasmid. So that's it for our tutorial, and we hope that you learned something from this video. Thank you for watching.